Welcome back. In our last lecture of this module, we're going to talk about MHC inheritance and this concept of HLA matching. We talked a lot in the last lecture about the genetics of MHC, but of course, with genetics, we have to think about inheritance. How do we accumulate all of the different versions of MHC that we have in our genomes? It's pretty typical, it's pretty standard for what you might expect based on any previous coursework in genetics, but a main idea that I want to stress here is that we inherit collections of MHC alleles that we call haplotypes. So a haplotype is a group of genes that is inherited together from a single parent. So remember that any individual has two chromosomes. Each of those chromosomes is going to have some combination of different MHC alleles on it, and so there's a whole bunch of them, remember, whatever that complement, whatever that combination of MHC genes is on that chromosome, we refer to as a haplotype. So there are many, a huge number of different possible haplotypes, but each person has two, which corresponds to each of their chromosomes. And so when a person is born, they inherit, or when they're conceived, they inherit two chromosomes, one from each parent. And so they're going to inherit one of either of the haplotypes that their male parent possesses, and then one of either of the haplotypes that their female parent possesses, usually. If you do this kind of in a Punnett square, you can sort of imagine there are four possible combinations then. If we call the two haplotypes from the male parent A and B, and the two haplotypes from the female parents C and D, then usually you're going to have most frequently one of four possible combinations: AC, AD, BC, or BD. However, remember that during the process of meiosis, when you're making gametes, you have rare genetic recombination events. And so every once in a while you get different pieces of chromosomes being recombined. And so sometimes we get rare recombination events like what you see here. So in this case, instead of inheriting either C or D from the female parent, the egg cell here made a chromosome which was a recombined version of a part of C and a part of D. And so the new name that we could give this arbitrarily here is R. And so this doesn't happen very frequently, but sometimes it does. And this is an additional source of diversity in the overall genetic, at the population level, the overall genetic pool of possible MHC haplotypes. So kind of the mixes are always being sort of refreshed every once in a while by genetic recombination events during meiosis. So if we look at this with a slightly deeper eye for detail here, we can see that the frequencies with which things recombine get a little bit more complicated because there are so many different possible MHC alleles. So here now, again, we have, parent A and parent B, we'll call them. Here we'll focus on MHC class 1 genes. So, remember that there are between 1 and 2,000 different possible alleles for most of all of the MHC class 1 genes. So A, B, and C. So here we'll say that, this parent on one chromosome has allele number 1 for A, allele number 37 for B, and allele number 3 for C. These are all completely random. And then on their other chromosome, they have this number, these particular alleles here. Same thing for parent 2. So for each of their MHC class 1 isoforms, they have the following allele numbers. And so what you might expect, or what you would expect based on these alleles from either parents, is that there would be four possible combinations. So all of their offspring would have a roughly equal probability of being one of four different combinations then. These would be basically this chromosome with this chromosome, which was what you see here, this chromosome with this one, what you see here, and then vice versa. So each of these four possibilities is one paternal chromosome and one maternal chromosome. But overall then, the overall combination of alleles that each of these progeny has is pretty distinct from either of their parents, because there's so much diversity even within individuals, is that when you combine even a random subset from one parent with the other, the overall or the complement of alleles that each offspring has is going to be at least 50% different from each of their parents, essentially. Um, and so this is one way that we make sure that we have a lot of genetic diversity at MHC, and it maintains a healthy overall population immunity within human groups, because each individual person is able to resist most infections that they see because they have such a wide coverage of different antigens. But again, remember that these are not the only possible combinations. Rarely we'll have genetic recombination events during meiosis, either in the generation of sperm or of egg. And so some possibilities of some of these rare recombined haplotypes that a person might, or in this case, the offspring of this couple might inherit, would be, for example, a haplotype which is A1, B40, C4. So this would be a combination of the two chromosomes that this parent had. So you can see here that they got A1 from this chromosome, but then there was some kind of recombination event that happened, and so A1 was combined with B40 and C4. So this recombined haplotype is going to take some of the MHC genes from one chromosome and combine them with some of the other MHC genes on the other chromosome. Same thing here. From this parent, we might have a possible recombined haplotype that is A3 and B5, but with C5 instead of C2, because a recombination event happened here, essentially. So again, these happen with different frequencies. There are a lot of complex rules that govern what is likely to be recombined and what is not. That's the stuff of genetics courses. But just keep in mind that all this system is pretty predictive of what we can expect from what offspring would inherit from their parents. It's not absolutely perfect because recombined haplotypes do happen. I bring this home because we often use the haplotypes of people for forensic purposes. So the haplotypes of the HLA complex are a frequent source of information for things like paternity testing or identifying an unknown corpse, for example, if at a crime scene. So we can look at an example of this for paternity testing, for example. So if we had an offspring, a random person whose parentage we wanted to determine, we could look specifically at their MHC1 alleles. So we can see this person, for their HLA-A, they have 3 and 43. For B, they have 54 and 59. And for C, they have 5 and 8. So obviously, we usually know who the biological mother of a person is in these cases, and we can take their blood and test their HLA haplotypes. And so immediately we know that whatever the offspring has, it had to have inherited from one or the other of its parents. And so if we look at the haplotypes of the mother in this case, we can see that she has A3 and A11. So if we look here, we know that the offspring had to inherit A3 from the mother, because they don't have A11. So if they didn't inherit 11, they had to have inherited 3 because they got one chromosome or the other, essentially. Same thing here. In this case, we can see that the offspring had to have inherited 59 from the mother, and here they had to have inherited 8. Because that's all that the mother had to offer, basically. So which of these possible 3 candidates then are the father of this offspring? Well, we have to sort of cross-compare now, and this actually isn't too hard to do. And in this example, it's pretty easy, actually, if we just look at the HLA-A isoform. So we said that this offspring had to have gotten A3 from mom, because 43, she doesn't have 43 to give. So that means that the 43 allele had to have come from the father. So if we look at all of the possible fathers here, we see that only one has the 43 allele. So we can pretty easily detect that potential father number 2 is the actual parent in this case. If we look at the other HLAs, we see a similar thing though. So mom gave 59, which means 54 had to come from dad, and that if we just kind of do an internal logic check, we see that dad did have a 54 to provide. Notice though that so did one of the other candidates. So 54 alone would not have been sufficient to identify the father in this case. Same thing here. The offspring had to get C5 from dad, but both of these potential candidates had C5 to give. And so C5 alone would not have been sufficient. So A here is the only possible case where there's a unique allele that the offspring has that only one father candidate has. And so A is the most informative allele here. But you can imagine, these are kind of like puzzles. Sometimes it's not quite so clear cut, and we have to do a little bit more kind of logical work to determine which possible combinations could have come from which possible parent. And so this is a very frequent way of testing paternity. But again, as I said, identifying people whose identities are unknown, and all we have are genetic information to identify them. And so HLA alleles, because they're polygenic and polymorphic, again, each individual person is going to have a pretty unique combination of all those possibilities. And so in a way, we can use the HLA complex kind of as a genetic fingerprint, if you will, to help identify people. The other reason that we often think a lot about the inheritance of MHC or HLA in the case of humans is because it's really important for a lot of medical applications, and in particular for tissue transplant and grafting. So let's talk about some of the different types of transplant because this helps us sort of put all of this into context. So, if you, for example, you had a very bad burn somewhere on your body, you might graft skin from a healthy part of your body onto the place where you had the burn, and so that you can sort of regenerate healthy skin in that area. When you move tissue around on an individual person, we call that an autograft. So remember, auto means self, and so a graft from self to self is what we call an autograft. And so using a mouse as an example here, that would be the case. However, we might that might not work for whatever reason, and so in some cases we might need to take that tissue from another individual. And so the type of individual that we take it from is going to determine what kind of graft we're dealing with. So if we take a graft from an animal which is genetically identical to the donor, we call that an isograft. Remember, iso means the same, basically. So an isograft comes from a genetically identical donor. That donor we could refer to as syngeneic. A syngeneic donor is genetically identical. Now, in mice, this is pretty easy to do because we have whole colonies of genetically identical animals through many, many generations of inbreeding. In humans, this is much harder to accomplish. The only syngeneic donor that you would have in any likelihood would be an identical twin, which very few people have. But so an identical twin would be a syngeneic donor, but frequently one of those is not available for human patients. In that case, we usually have to source a graft from a genetically distinct donor, and we call those an allogenic donor. And so tissue that comes from an allogenic donor we call an allograft. And the idea is that we need to get that person to be as genetically close to the recipient as possible, but there's always going to be some difference, and that difference is usually the source of most of the complication with graft rejection, as we'll see. Another possibility that's becoming more and more common with technological advancement are what we call xenograft, which is where we actually take tissue from a different species. Now, frequently, different species are very genetically distinct from the recipient, and so there are a lot of things we have to do to make this work. But you should at least be familiar with the name. If we have a graft from a different species, we call that a xenograft. So let's talk about some of the issues with grafting as they relate to MHC. A lot of this work has been done in mice, and so I want to introduce you to, again, sort of the nomenclature of how we talk about mouse MHC, particularly with inheritance. But let's imagine that we have two mice, and they are homozygous for the same MHC haplotypes. So for this mouse, we'll just call it B. So B just stands for all of its MHC genes in combination, and so that particular combination of genes, we just refer to it as B. This mouse is homozygous, so it has all of the exact same alleles on both chromosomes. And so this is common with laboratory mice, again, because they're frequently very inbred. So this mouse we'll call H2B. So remember, the mouse MHC locus is called H2. Its haplotypes are B. It's homozygous B. the second mouse that we're going to use in this mating, it's homozygous for a haplotype that we'll call K. So if we mate these two animals together, again, we're going to get one chromosome from this mouse, one chromosome from this mouse, and so the resulting progeny, the F1 cross in this case, is going to be heterozygous. It's going to have one chromosome that has the B haplotype and one chromosome that has the K haplotype. So what does this mean in terms of if we wanted to sort of within this mouse family do some tissue graft experiments, or if we wanted to do grafting for some sort of scientific purpose? Well, as we'll see, it really matters quite a lot to which mouse is able to donate tissue to which recipient. So if we take, for example, the original BB parent, the homozygous B parent, that parent could donate a skin graft to another BB mouse, so perhaps a sibling mouse that had the same genotype as it did. But it would not be able to donate skin to the KK mouse, so the mouse that it was mated with. That's because they don't have the same MHC alleles. And as you'll learn in a lot more mechanistic detail later in the course, when mice have MHC alleles, or when mice are introduced to new MHC alleles that they did not have at birth, that frequently is the source of what we call immunological rejection, which in its sort of advanced stages becomes graft versus host disease. So we're not going to talk about exactly why it's rejected, but know that we sort of, when we're born, we learn which MHCs we have, and then if we see any new ones later in life, we reject those pretty firmly. So this KK mouse would reject a graft from a BB donor, essentially. However, a mouse that was heterozygous, so the progeny of this cross, the heterozygous mouse, it could receive a graft from a BB parent because it was born with B, and so new B tissue is going to just be more of the same. It's used to B, it knows that B is self, it recognizes B as self, and so it could take the graft. Even though it has K, basically this mouse develops what we call tolerance to both B and K because it's born with both, so it could take B, and as you can see here, it could also take a graft from the K parent, whereas the K parent could not donate tissue to the B mouse that it was mated with for the same reasons. But if we take that progeny mouse and we take skin from it, what we see is that the heterozygous BK mouse, it could donate skin to another heterozygous mouse, but it could not donate skin back to its parents. That's because its skin has both B and K, and so a mouse that is pure B, homozygous B, it's going to reject the K MHCs, and the mouse that was homozygous K is going to reject the B MHCs that are found within that heterozygous graft. And so this kind of thing is a very simple definition. Now, most of us are not homozygous for anything in our MHC complex, and so actual matching in terms of human donors is actually much harder than this. But this is a simple diagram to kind of show the principles at work. Basically, if a progeny has received some new MHC allele from one parent, frequently that means that donating from children back to their parents is hard because they have one chromosome from the other parent that is usually not compatible with the parent who needs the graft, and so that frequently doesn't work, as you can see here illustrated with this mouse example. So if we think about this in a more clinical context with actual people, what we see is that allograft success really requires careful HLA matching. So we need people to have as many of the same alleles as possible. And so, it's usually impossible to match all of the HLA alleles, again, unless you have an identical twin. So frequently, when we're trying to find donors for graft, whether it's skin or whether it's kidney in this example or whatever else, you usually just have to settle for as many matches as you possibly can. And so what this graph shows is an example of this. So this is focused on three MHC alleles, or three MHC isoforms, HLA-A and B, which are MHC1, as well as DR, which is one of the MHC2s. So remember that we're talking about three different genes here. You're going to have two alleles for both, so there are 6 possible genes that we're dealing with here. So you have one allele for or 2 alleles for HLA-A, 2 alleles for B, and 2 alleles for DR. So as you can see here, you could have anywhere between 0 and 6 mismatches, which is what MM stands for. So this is plotting survival following the graft, and on the y-axis here. And so we all started 100. So following a graft, it follows out patients who received a kidney transplant from a donor who had the indicated number of mismatches in these 3 HLAs. And then on the x-axis is time. And so what you can see is that patients who received a graft from a person with no mismatches, so perhaps an identical twin, or in this case, somebody who at least had identical alleles for all 3 of these HLAs, they do the best. So if we follow their survival over time, we see that by the end of year 3, still something like 87-ish% of them, we still have graft survival at that time. Whereas patients who had 6 mismatches, so they were not matched on any of these 6 alleles, they had the worst prognosis overall. They have only about 78 or 80-ish% survival after year 3, and you can see it's a pretty a linear-ish correlation here. The more mismatches we have, overall, the worse outcomes we have in terms of graft survival. Again, why exactly this happens on a molecular level is the topic of a future lecture. But for now, what I really want you to appreciate is that these issues of inheritance are important clinically because they determine what kind of options we have, not only for tissue grafts and that kind of thing, but also things like blood transfusions and so on. We need to carefully match these immunological parameters across tissues to make sure that they're not rejected by the recipient. One additional thing that I'll say about HLA alleles is that we can also use them kind of as a genetic fingerprint of actual populations of people. So the frequency of certain HLA alleles are really concentrated within either geographic or demographic, ethnographic populations of people. So for example, the HLA-C2 allele, we see it most frequently in people of whose origins are in Africa, and then the frequency of that allele is progressively less the further away from Africa we get on the continent. Now, obviously, in modern times, we've had so much population mixing due to modern transportation technology and the history of colonialism and all those sorts of things. And so, usually, what these allele frequencies tell us is more about where a person comes from genetically, more than where they happen to live right now. But again, this allows us to study things like human evolution and the migration of early human populations across the continents, because these allele frequencies are found, certain MHC alleles are found with really high frequencies within really genetically distinct populations of people. And so this is another common tool in science that we use that's unrelated actually to immunology. In this case, again, it's just the identification of people, and this allows us to do a lot of additional cool research at the population level. Okay, let's summarize MHC inheritance. MHC genes are inherited as haplotypes. Remember, the haplotype is all of the genes that you inherit on one chromosome from one parent. A caveat to that is that sometimes rare recombination events, which can result, which can result in mixed haplotypes. And so you might actually inherit genetic material that is derived from both of the chromosomes from a single parent. In any case, MHC haplotypes are useful genetic markers of identity, so they're frequently used for things like paternity testing, forensic analysis, etc. Remember that many alleles appear with high frequency within certain geographic or demographic groups, so we can use it for population level genetics as well. And finally, HLA matching is required for tissue grafting. Non-self MHC causes an immunological rejection response, and so we need to try to match our HLA alleles as closely as possible when we're doing tissue grafting. Isografts are best. Remember, isografts are those that come from syngeneic donors. In humans, these would be identical twins. But without one, we have to rely on allografts typically, and so we want that allogenic donor, again, to be as close to us genetically at the HLA complex as possible. Okay, that will finish our discussion of MHC and antigen presentation and finish out this module. So the next exam will cover all of the material in modules 4, 5, and 6. This should say summary 6.4, by the way. This is the end of module 6. Okay, so that is the end of this section of the course. We'll do some more review, obviously, on our Wednesday session. I'll post a study guide, but let me know if you have more questions via email if you have them. Thanks.